All right, Sarah, you're ready? Okay, let's get started. So everyone, welcome to today's Taos seminar. It's a great pleasure having Sarah Brooks here from Texas A&M, the Atmospheric Science Department, where she's professor and director of the Center of Atmospheric Chemistry and the Environment, which deals with air quality, health-related issue, how climate and air quality are interlinked in a way. And before we start with the talk, a quick reminder, please maybe mute your microphones. There are many who have their microphones still on. It is recording right now, and I introduce Sarah. A few words. Sarah got did a bachelor in chemistry at MIT. Then she went into, this I didn't know, analytical chemistry, got a PhD in analytical chemistry in the University of Colorado. Afterwards, she did a postdoc in atmospheric science at Colorado State University. And then afterwards, she became faculty at Texas A&M. She received numerous awards, so just a few highlights here. She is a Career Award recipient, and she has the Presidential Early Career Award in Science and Engineering awarded by the White House. That's a very special honor. Of course, several internal and university-wide awards like the Texas A&M Impact Fellowship. So Sarah and I just try to recall our past, and I can say this year, I think we know each other by 20 years or something like that. And I think what happened as Sarah graduated, or in the year where she graduated, she came to a European Aerosol conference in Europe, and that was in Germany, visited Switzerland, where I did my graduate studies in Zurich at the ETH, and visited my lab. I still remember that, she coming in the lab, and we are talking aerosols and nucleation. And then to our surprise, the last 20 years, our topics crossed several times, from organic aerosols, marine aerosols, a topic today. And so it's great and long overdue having you here, Sarah. So I'm very pleased to have you. And with that, we get started. Sarah's talk is Unraveling Mechanisms Underlying Plankton Blooms and Their Implications for Biogenic Aerosol Properties and Cloud Formation. Please take it from here.
Hey, thanks, Daniel. This is absolutely as much fun as one can have on a Zoom meeting. So I'm really pleased to be with you all today. And thanks for that really nice introduction. And so today, let's see. I can move along. Oh. If in fact I can move forward. Yes, okay. So today I'm mostly going to speak about the NAMES project, which I was part of. This is a project in the North Atlantic Ocean, and I thought it might be interesting to both oceanographers and atmospheric scientists. For me, it was an opportunity to really appreciate how complex things in the ocean are, and how they may play some very important roles in both warm cloud formation and cold cloud formation, which will be the foci of my talk. And so perhaps no one here needs to appreciate how complex the biological diversity is in the ocean, and that's something in atmospheric aerosols. We're just starting to appreciate biological diversity, I believe. And so if we look at it here, first of all, you can see in the MODIS image how we have a phytoplankton bloom in the various colors of green, and you can imagine the dynamics and the eddies of this. And then overlaid on top of that, we have clouds. And so these are the things that we are thinking about in conjunction with one another. And then if you look in panel B, a coccolithophore, this is one of my favorite pieces of biology. Just look at all the plates on it. Of course, we wonder, could this be an ice nucleus? Get to this later. And then in C, we have a picture of some representative diatoms. Okay, so while there is a wonderful amount of, so I would say that NAMES is a resource at this point. There's a wonderful amount of ecological and biological information that was collected during this project. So NAMES stands for the North Atlantic Aerosols and Marine Ecosystems Study. And it's what's called an Earth Venture project. And so NASA provided the opportunity for us to go out and do 4 measurement sampling campaigns by ship in the process of 5 years. At the same time, there were flight measurements, and there were gliders and drifters, and a serious amount of modeling. And so overall, this was a really big project, all under the auspice of trying to provide information to inform satellites. So the NAMES data is now being used to interpret satellite data. All right, so of course I can't cover all of NAMES today. And so I will discuss specifically marine aerosols and how they can be catalysts. Don't worry, it's not a chemistry talk, but they can be catalysts, or perhaps you like to say seeds, for both warm clouds, that is by acting as cloud condensation nuclei, or they can act as ice nucleating particles. If time permits, I will then discuss some laboratory measurements that are really inspired by the NAMES field data. Okay, and so here we are in NAMES. You can see the RV Tom Thomson, which we took from Woods Hole out into the North Atlantic Ocean multiple times. The goal here from the ecological perspective was to sample a phytoplankton bloom in the summer and winter seasons, and to evaluate the summer-winter differences and what the initiation of a bloom really, what drives the initiation of blooms. And so we had the opportunity to do that. And then more related to my focus, when and where are marine sources of cloud-forming aerosols really happening, or where are they observed? And so I will be discussing briefly today some CCN measurements that were collected both from the ship in order to tease out detailed information on things that might be cloud formers. We also collected sea surface microlayer samples and looked at their cloud forming properties. And we also, with Rich Moore, had another CCN instrument on board the NASA C-130 aircraft. And so we took a good look at that. Okay, and so this is this is the study region, and this is the nominal campaign plan. So if you've been part of a large project, you learn that the best plans are made, and then they do change a bit, but largely for the most part, we kept something like this. So we started in Woods Hole with the ship, whereas the aircraft starts here at St. John's for easy access to this North Atlantic sampling region. And so the ship goes full stream ahead out to somewhere at about 40 north, 40 west, and then chooses to stop. And from the ecological and ocean perspective, most of the exciting measurements are done by CTD. So by having the opportunity to stop and collect water at multiple depths. And so there are a number of stations along this transect. And of course, in the course of 5 years from 2015, spread over to 2020, there were some changes in what we did, but this is about where we were in this general area, and we did get lots of information in that area. Okay, so aerosols over the North Atlantic Ocean are a complex menagerie of things. First of all, you have ocean sources. So many of you will be familiar with the claw hypothesis from 1987, where the idea was that phytoplankton produced EMS, produced aerosols. this was long thought to be the only way in which aerosols are sourced by the ocean, or at least marine aerosols are sourced by the ocean. But in more recent times, we've also appreciated bubble bursting mechanisms, which could lead to, well, which could provide an opportunity for organic rich films or material from the sea surface microlayer and perhaps larger organic molecules to also be enhanced in concentration and then injected into the atmosphere. And they could be both cloud drop formers or ice formers. And of course, at the same time, this whole situation is quite complicated by transported aerosol from both other marine locations and from continental sources. Okay, so this is not a talk about the CLA hypothesis, but this is one of the many insights that we got by having the great resources of names and many measurements and measurements in multiple seasons. And so this is, in short, the CLA Saga, as I call it, goes like this. So you may know that the CLA hypothesis was proposed in 1987 by four individuals whose name give us CLA. So Carlson, Lovelock, Andrea, and Warren. And this was really proposed as a link between marine biota and climate. And the basic idea was that DMS acts as a gas phase precursor, which provides our sulfate aerosol, which can be CCN, and which can then produce clouds, playing into cloud albedo, and so forth. Okay, so that was a great idea, hypothesis, and it still is, but many years went by where poor experimentalists like myself tried to confirm the CLA hypothesis. And sometimes there was evidence of the CLA hypothesis, and sometimes there was not compelling evidence in the field data. And ultimately, this led Trish Quinn to boldly write the case against, pardon the typo, against climate regulation by, this is a mouthful, via oceanic phytoplankton sulfur emissions. In short, she wrote that the CLA hypothesis is dead. You haven't seen it for all these years. So what in the world is it? And it must just not work at all. Since then, more analyses were done, and now we get to enter names. I call this the zombie CLA. At this phase, we'll see, and I'll explain some more details, that CLA isn't completely dead. It's not completely alive either, and it would be no surprise to aerosol scientists that, in fact, the situation is just more complicated than this simple and elegant hypothesis controlling the whole show. Okay. All right, so since I'm going to talk about cloud formation a bit here, let me just go back to extreme basics for one moment. So here I have a Kohler curve. And what you need to know if you're not a cloud microphysicist is this. If you know an aerosol's composition and size, we have a great theory here. You can predict with great reliability whether it will form a cloud or not, and at what critical supersaturation the ambient environment must reach for cloud formation to happen. Now, conversely, if you know the composition of your aerosol, that doesn't mean you know, if you know it's CCN, you don't necessarily know its composition. And that is because multiple compositions of aerosols might very well have similar CCN properties. So if you really want to understand what's going on out there, and we certainly do, what do? Okay, we measure all the properties. We measure size and composition and CCN activation, all in the same project. Okay. And so here is a picture of us mobilizing. This is part of the NAMES crew, mostly the graduate students I had on this project. And here we have Lynn Russell and some of her graduate students as well. We are here's my student Alyssa installing an inlet on the front bow of the Tom Thompson, and we sampled with Lynn Russell through this inlet. There was an identical aerosol inlet in a second fan beside that, led by Trish Quinn and the PMEL group. And so we really do have a wealth of measurements to look at if that's something that you want, and they're publicly available in the NAMES database. Okay, so here we have our CCN instrumentation. My student Brianna is showing it off. She's quite pleased about the fact that this condensation particle counter is now working correctly. And so here we had counts of particles, particle sizing with the differential mobility analyzer. Unfortunately, the CCN is kind of cut off. You can see it starts here. So we had continuous CCN sampling. And then for this particular project, the ice nucleation was only done offline in my laboratory. And so here we have some Pixie sample. This is a cascade impactor to just collect aerosols in appropriate size, in size resolved, swaths of size resolved, supermicron, submicron aerosol, and take them back to the lab for analysis. Meanwhile, we also collected with a low-tech screen technology, which is maybe the standard for sea surface microlayer collection. We collected the sea surface itself, and I will show some measurements that we did on CCN measurements that we did on that screen sample. Okay, so I'm going to jump to one really important thing that we learned from this culmination of CCN measurements and the fact that we had 2 winter and 2 spring campaigns. What we did learn is that a significant seasonally variable fraction of aerosols arise from the local sea spray aerosol mode, which can be attributed to DMS. So overall, in the wintertime, in the North Atlantic, 50% of the total aerosol mass is related to DMS or was on these trips. And in the other campaigns in the fall and spring, there was still significant evidence of this, but it was less than 20%. So in terms of CLA, there's a very nice summary that I won't provide that Chris Quinn led that. It's one of the many NAMES papers. And the take-home message here is that, in fact, the CLA process does produce cloud-forming aerosol. So as a process, that is a process that works. However, it is not the majority of the aerosol responsible for cloud formation. Okay, and so I'll leave that there. This leaves us wanting to understand those other CCN potential sources, like the primary aerosol, and what in the world is happening in the sea surface microlayer. And that gets us to the biology. What is it that might produce additional CCN, and under what conditions? So when you want to compare the cloud forming properties of different aerosol populations, often we take the Kohler theory and we modify it and simplify ourselves to a popular analysis called the kappa analysis. This is a single parameter which is meant to account for chemical effects on cloud activation. And if your samples are internally mixed and all completely soluble, kappa works fantastically well. And so that's what we're presuming here. And so there are 2 things we can do. We can calculate kappa directly from the CCN measurements, which we're doing on the ship. And then I must say the same instrument comes home, we aerosolize the CCN, and we aerosolize the sea surface microlayer samples and run them through the CCN instrument. So that's a direct measurement of critical supersaturation and a direct calculation of kappa. Okay, there's an alternative thing that we can do as well. If you have no CCN direct measurements, you can calculate kappa using, well, you can predict it using some other measurements available on the ship. And so then we use the aerosol size and chemical composition. So if we do kappa both ways, this is going to allow us to test our ideas about which compositions are having an influence on that cloud droplet activation. And just for comparison, here's some nice work earlier work by Marcus Petters. And you can see here we have the dry diameter critical supersaturation. And you can see how kappa values are related to clouds of various compositions. There are many compositions on this graph. What we need to appreciate is there are a number of organic compounds and there are inorganic compounds. And so the ammonium nitrate, the ammonia is down here, okay? And so you can see that when you have a higher kappa value, like out here in the, okay, so out here we have the higher kappa values. We can have, I'm sorry, here's our kappa values. That's much better. All right, so here's our ammonium nitrate, much better. So we have inorganics on this end, and we have high kappa values. And as we add these organics, we will in fact reduce the predicted hydroscopicity factor. Okay, so this is well known and it checks out in a laboratory situation. Let's go to our NASA NAMES project here. And the samples in this study are really collected at times when the screen sampling of sea surface microlayer was on the docket as something that we did. And so here we have Woods Hole, and these are points on the transect where the sampling was done. Names 3 and 4 were included to something that occurred to us after names 2 as a great idea. And so I'm pleased that we did add it. And so we only have names 3 and 4 data, and we have winter and spring. Okay. And so what we did as an experiment, we collect some samples, and we want to really understand the organic component, what's going on with the organics. And so we took some of the samples and desalinated them to remove the salts, because the salts are really, there's of course so much sea salt present. It's convoluting what's going on. So how can we learn what's there with the salts present as well? And so then we take both the fresh sea surface microlayer and the desalted samples and produce some aerosol and run some measurements on them. So here is once again the basically direct measurement of the kappa value. So we have a differential mobility analyzer and a condensation particle counter. This is going to give us a specific size of particles, which we then vary. And our CCN instrument is going to tell us first which size is coming in. For any size coming in, what supersaturation is required to reach that critical supersaturation to be able to grow. So we can measure these kappa values. Meanwhile, we wanted to do some measurements to provide the inputs constraining that information. So the surface tension is measured with the tensiometer. We have chemical composition, and of course we also have size. And so these are the inputs to the predicted kappa value, wherein you don't have a direct CCN measurement. Okay, and so with that, what we see in this chart, we have the critical diameters and the kappa values of aerosols, and they're from a number of stations, and you can think of the rainbow colors here as the SML, and we'll consider them as a group, and then the open circles and other symbols, those are the desalinated symbols. And so all in all, what we see is that desalination increases the critical diameter, so it makes it harder to form a cloud drop, and it reduces the kappa value. And probably the most important thing to appreciate here is that really in your actual ocean sample, where you haven't desalinated it, the CCN activity is really being driven by the salt. This could give us some sort of comfort if we are spending years trying to understand the impact of the organic. The impact of the organic is potentially not very large, considering all the salt that's out there in the ocean. Okay, well, it was good to know that in detail. Now, but we still wanted to finalize what amongst possible biologically derived organics in that sea surface layer might be having the impact that is there, albeit small. And so we chose, these are the 6 compounds that we chose as potential components of the sea surface microlayer. And you can see we chose, well, chlorophyll because there we are in the ocean, rubisco as a representative enzyme that is part of photosystem 2 and is very prevalent in the marine environment, glucose, humic acid, fulvic acid, and trilene. And some of them worked better than others. And in fact, I'm only showing 3 in this final chart. So what we did with those compositions is we said, okay, imagine that's the composition. I know the molecular weight and density, put that into my, go ahead and use that to predict a kappa and compare to the measured kappa values. And there is some uncertainty in this. The desalination process is not perfect. And also there, as the oceanographers here are well aware, you have maybe 20 most important sea salts. And so this is arranged to represent the high and low values of salt alone. That's just as a backdrop. And what we see here in blue, we have our collected data. In green, we have glucose, so what the predictions based on glucose. Then in blue, we have humic acid, and in red, we have the rubisco. And so it's difficult to see in the native samples where all the salt is still there, really any impact. So it's fortunate that we did desalinate the samples to look carefully at only those organic, only the organic when it's not overshadowed by the salt situation. And so I hope you can see in the bottom 3 panels that of these choices, the large molecular weight rubisco is the best if you were to choose a single component. And a lot more work needs to be done here, but what it does show you is that the primary aerosol, assuming we can get this sort of primary aerosol out of the sea surface microlayer and into the aerosols, then this very large primary aerosol would be representative of the influence that it has. Okay, so I will just conclude for that piece. So first of all, we generated these aerosols and desalinated some of them for comparison. The take-home message is not always super exciting, but it helps to provide insight. And so I guess baby steps along the way. The CCN activity of the SML was largely driven by the salt, okay, as evidenced by the fact that the desalinization drastically reduced the kappa. So this says that the organics, we extrapolate this to imply that the organics did not enhance cloud formation. And so they are not going to be the major player, which potentially is part of why resolving the cloud hypothesis and the marine influence in general was so difficult, because it takes a lot of organic to have any influence at all on your cloud formation. And so that's that. For the influence that is there, it is best represented amongst the simple things that we looked at by rubisco. Okay, so by larger enzymes and other proteins and other larger components of marine life. Okay, so in short, in terms of warm cloud formation, this was it. We during names learned some nice details, and we also, in broad strokes, looked to see that some claw hypothesis, so some SOA is there, some primary aerosol is there, and we really have a pretty good understanding now of the aerosol warm droplet situation. So maybe that's, if there are grad students listening along here, maybe CCN of marine aerosol is a difficult thing to now write a proposal on. So maybe you should move to the ice phase, which is near and dear to my heart. So we're going to now shift over to ice nucleation. In case you're not already in love with ice nucleation, apologies, there are so many things on this IPCC graph, but I bring this up because I want you to just appreciate how much ice we have at all the latitudes, okay? And so please go ahead and see we have distributions of cloud in A, and then this is a marine cloud contributions. I'm sorry, this is not all about marine cloud contributions, but the marine clouds that we're sampling in names are going to be here in, about 40 degrees north, and in this region, and in fact across all of these latitudes. I just want you to appreciate that when we look at the total water path, a huge fraction of this is ice, and then naturally that's across all latitude, or excuse me, across all elevations. But indeed, this ice is going to be a really important role. And so I'm coming from Texas, and I have to remind students that it's not just interesting and exciting in the polar regions, which of course it is, but in fact, certainly in the North Atlantic and even in the tropics, we need to not forget that ice can be a significant part of the equation. Okay, so many things can be ice nuclei or ice nucleating particles. And in the past 20 years, there have been many measurements on sulfates, organics, and dusts and soots and so forth. And in more recent times, but still maybe last 5 to 10 years, we have looked at sea spray and biological aerosols. And we as a community, certainly not just I, have looked at many types of biological materials, and have even looked at viscous organic liquids, which could in fact be a contact ice nuclei, among other things, or an immersion ice nuclei. But I will save that for another day. Just to look at the ocean again, here we have the sea surface, and here we have the sea surface microlayer. And there are many mechanisms for ice nucleation, although the most popular and in the troposphere under most conditions, the most common mechanism is the immersion freezing. And so an aerosol is released to the atmosphere. It manages to engulf an ice nucleating particle, which later cools and freezes. Okay, and if you are starting in ice nucleation, perhaps you start there. So for the purposes of this talk, we're only going to talk about immersion. Okay, now back to the names. So again, this is just a highlight of an interesting thing that happened in names. And in fact, I'm just going to show data from sort of an event of opportunity. So for the names 2 projects, here are the stations, and something very exciting happened before we went to station 5. Right before station 5, there was a storm, and it disrupted the depth profile. It totally mixed the layers in the ocean, and so we didn't have the conventional structure in terms of temperature or chlorophyll in that ocean. And so we had an opportunity to sample under these kind of strange conditions, where here we have 0 and all the way down to 200 meters, we have a well-mixed layer. So, and the chlorophyll is also basically uniform across those. So eventually, by the 4th day, you can see that near the surface, you have a lot more chlorophyll, and the temperature is warmer as well. And I think it's safe to say that this is more of a typical situation when you haven't just had a storm. And so typically, you would have the opportunity for chlorophyll and anything associated with it to be injected into the atmosphere. So this was exciting because the storm basically subtracted the organics from the situation. And we were able to stay on site and watch as that was reset up. Okay, so now yet another nice thing that happened during names was that Timothy Bates and Trish Quinn brought their sea sweep. So this is yet another type of sampler. And here we have, it's basically, it's on a nice, it's like a nice kayak, right? So you can see a catamaran and a giant vacuum, more or less. So this is a sealed container floating on the sea surface, and air is pumped into that to artificially produce a headspace of aerosols, which we can then vacuum up into the inlet you saw earlier, and all the way to the Pixie impactors. And then to do the ice nucleation, it was necessary to keep things in a -80 freezer and then bring them back home to Texas A&M. Okay. So at Texas A&M, we conducted measurements with a very simple technique I've been using for quite some time. This is our ice microscope apparatus. We have just a microscope here, and we have a controlled environment. So we mix dry air and moist air to control the relative humidity over a droplet sample. And inside a sealed Lincoln stage, we have a droplet, and then so if we're doing these field samples, we take our Pixie impactor sampler, we put it right here onto the slide, and then we repeat freeze and thaw cycles and watch freezing and cooling and freezing and cooling. In the names case, it's actually only one drop and one sample each time. And so we did have duplicates. We had 2 samples every time we went out. That's not a lot of samples. This is something to be aware of. We now have the opportunity, we're now routinely sampling 16 samples, still not an enormous number, but doing it this way does allow us to get some very detailed information and to take a close look at what's going on. And so we can do, so for example, we can look at immersion freezing, which is what we will do today. But if we were, and we have in other studies, if we were to take our ice nucleating particle, now granted, this is a particle that we have sieved and placed adjacent to the droplet. So this is not from a Pixie impactor stage. But in this case, we can do contact freezing. I think this is very fascinating because by virtue of position at the surface, rather than in the bulk of the droplet, we will warm the freezing temperature by about 4 degrees, regardless of the type or composition of the INP. This is something that doesn't get a lot of play in the atmosphere because, admittedly, in the atmosphere, you have to first have the situation where your droplet and your INP have collided and are in contact together. And so probably the rate limiting step here is the collision. Nevertheless, there it is. Okay, so we're going to focus on immersion. And here we have the mean nucleation temperatures of these Pixie impactor samples. And they're plotted against the station data. So this is just measurements collected on station through the sea sweep and the Pixies. And highlighted, we have the 4 days of that storm of opportunity. And what we can see, these are what I call moderate ice nucleation temperatures. So I should point out that somewhere down here, at minus 38, or in my apparatus, more like minus 34. We have homogeneous freezing of droplets. Okay, and I say in my apparatus simply because the substrate that we're on doesn't get us all the way to minus 38, and in fact the droplets are a little bit big. All right, point is, if you're thinking about this data, any freezing temperature above this is something we would classify as an INP, or an ice nucleating particle. Of course, these are moderate INP. They're not that impressive. If we had a really good ice nucleating particle, we could see nucleation at minus 15 or 10, or even in some biological cases, much closer to 0 Celsius. Okay, but here we see we have nucleation, and what I want to appreciate, and granted this is only 4 days, but the nucleation temperature got much warmer on the last day. So we think that's interesting, and we want to understand why. So now, just for a different look, we thought we would compare the ice nucleation to phytoplankton biomass. And so we've plotted the fraction of droplets frozen by minus 25 or warmer. In other words, how many of the droplets contain moderate INP. And then in this, you really see that that's extremely the case just on that day 4. Okay, and that is, keep in mind, when the microlayer has resolved itself and you have organics to sample from, and that would be primary. If that is what's going on, then we would have primary organic aerosols as INP. Well, here's the surface, or near a surface as we can get, chlorophyll concentration. Of course, we had hoped that this chlorophyll concentration might explain this high INP, and that is just absolutely not the case. So hope is useful in science, but only if you keep it in check. That would be a hypothesis in which all of the organic material was the same, and that was probably too much to hope for. So we thought we should look more carefully at individual things that contribute to the chlorophyll, or individual types of organic. Wow, and I have 2 the same color. I apologize. We thought we need to look better. We need a better approach than just the kitchen sink. And so instead of looking at everything that fluoresces as chlorophyll, what if we were able to select different sizes of particles from the sea surface microlayer, or the upper levels of that, and then measure their ice nucleation ability? And so fortunately, Mike Barenfeld and Jason Graff were on board, focusing on various organisms, and I do apologize. The pico-eukaryotes are supposed to be it doesn't look like orange at all there. I'm sorry about that. So what we did, or with their help, is collect surface waters and send them through the sorting flow cytometer. So then the convenient thing, and perhaps marine biologists are very familiar with this, is that we can nominally choose organism categories broadly by their size. And so we have some Synechococcus particles. They are particles that are half a micron up to a micron. The nanoeukaryotes are going to be much larger, 3 to 50 microns. And then the picos, I'm sorry, I'm sorry, these are much bigger. And then the picos are in between, these are out of order, 1 to 3 microns. And so if you look at what these all do, and so first of all, this is just Jason's effort to quantify the reorganization of material in the upper layers of the ocean. And sure enough, the overall chlorophyll concentrates at the surface by day 4. We saw that earlier. And here we see each of these types of organisms also are increased at the surface by day 4. Okay, well, that doesn't tell us which of these they may be, but there they are. So let's then take and see, so they're all potential candidates as for INP. And so we took direct seawater samples and measured their freezing or the nucleation temperatures in the ice microscope once again. And what you can see is they actually are quite poor. So in terms of the pico-eukaryotes, you're breaking over homogeneous freezing, but only just a little bit. And in fact, the nano-eukaryotes in all this salt water are even not freezing are freezing at colder temperatures than a pure water droplet. And so not much to write home about there. However, would you have all of this salt water in a cloud drop in the upper atmosphere? And so we say, well, no, we wouldn't. The first thing that would happen in the immersion freezing process is that our aerosol would become a droplet, and so we consider the size of a droplet that when it initially activates, and that would translate us to an atmospheric sodium chloride concentration way down here below 5 grams per liter, whereas the initial seawater is up at 30. And so this is a study of the same 3 organisms under a series of salinities. And you can see that at last, when you get to what actually represents the atmosphere, we now have, in all 3 cases, we have reasonably good moderate INP once again. So this says that, in fact, these could serve as ice nucleating particles. Okay, now granted, we don't know, we can't isolate a single compound, and of course there are so many others, but I think that provides a little bit of insight. Now, something else that we saw on this project was that the growth rates changed over the course of Station 4 as well. And so this is a plot calculated by Susan Menendor's group. And so I thank her for this plot. She has not yet published it. And so here we have increases in chlorophyll production, and she has adjusted for larger organisms eating smaller organisms. So the grazing, it's adjusted for grazing rate. And so what we see is this fast growth just before day 4. Okay, and what in the world could that mean? Well, okay, we don't know for certain, but there is this suggestion that the accelerated growth that happens, that provides and generates some INP that are then a day later improved INP. And again, that's a suggestion, and so we take it to the laboratory to think about it. But we have, as a summary of that data, we have small phytoplankton cells do catalyze ice nucleation. The Synechococcus is one of them, and it's worth pointing that out specifically because the Synechococcus is also the most abundant phytoplankton, and it is a functioning INP, so it could certainly contribute. It is important to keep an awareness of the fact that in concentrated seawater droplets, the salt inhibits ice nucleation. And this is an open question, this possible connection between accelerated phytoplankton growth and improved ability to act as an ice nuclei. And that is something that I don't have a ton of time to discuss, but I want to show you that we pursued that idea in laboratory conditions. And here we have 3 really different examples of potential biological INP. And what I will do is I will show you, so now we say, okay, we're in the laboratory. So the nice thing here is we have a MARC tank. So it's basically, we have a large fish tank. We're controlling the nutrients in the tank. We're controlling temperature and light, and we can monitor our culture. And then we can use a water falling droplet generator to produce aerosols in the headspace, and then sample, again with those pixie impactors, the ice nucleation off aerosols produced in these conditions. All I want you to appreciate here is in the beginning of a study, the growth rate is really high. This is not a surprise. If you look at the cell counts, it is common for a static tank like this one in which we put the nutrients in the beginning of the tank to increase in concentration in what we call an exponential phase, and then have a stationary phase. And okay, biology, it doesn't always do what I wish it would. This is not a stationary phase, but it's more level. And then there would be a phase of decay. And so as a consequence, of course, the growth rate is highest in the beginning of this study. All right, so we see this for the Thalassiosaurus westflagi. We also characterize, especially my collaborator, Daniel Thornton, did the biological analysis here. And so we see briefly that the physiological status of the organisms is strong in the beginning. We don't see any evidence of autocatalytic death until much later. And that's an aside. Cell death was certainly thought of as a potential IMP. We see no correlation there, which we'll get to. And then also because we had been interested in cell death, we had cytox staining to look for leakage of samples. And in the beginning, when the growth rate is high, the nucleus is intact. And we have many samples, and we have lots of unstained samples. So we're not leaking yet. And again, that would have been important if we were thinking about cell death. So there we are. And so what we see, and this is just a little bit of data, but we see that in black, that's Thalassiosaurus westflagi, freezing temperatures are in fact warmer when the growth rate is high. Okay, we did these other two very different types of organisms. The data for Emiliana Huxley leaves a lot to be desired, and we will repeat that. But what we see broadly is that in all these cases, when the organism is undergoing rapid growth, that's when we get the warmest freezing temperatures. And that is interesting. So it's also true in terms of ice INP concentrations. They are highest on the early days in the experiment, which are in these set of measurements over here. And so I will stop with a summary so far and remaining questions. So we see that the ocean is a source of moderately effective INP. So these do catalyze ice nucleation under, the 20 to 34, negative 20 to 34 Celsius region. In the mesocosm and in the North Atlantic Ocean, we see evidence of connections. And, again, I'm sharing with you things that we don't have fully resolved, but there is evidence that the organisms are undergoing rapid growth and are potentially better INP at that time. And so that makes us wonder, what in the world is a characteristic of their physiological state that might be different during rapid growth that could also that would cause the better ice nuclei. And I see that I've used absolutely all of my time. So I'll just say that from the NAMES project, we now have a really good understanding of marine CCN. And in contrast to that, in marine ice nucleation, what we have is a set of compelling questions and a need to do more work. And so thank you very much for your attention. And I'll stop there.
Thank you, Sarah, for this nice talk and overview of the NAMES campaign, and also highlighting, and what I very much appreciate, and colleagues here too, the multidisciplinary nature, or really linking the ocean and the atmosphere together. And there are still a lack of such studies. As a matter of fact, I open the floor for a few minutes of questions, and then as usual, the grad students can stay with Sarah for a little bit of discussion. Are there any questions? Please go ahead. Ernie Lewis, yes, please. You can unmute yourself.
Okay, Sarah, thank you. That was a very interesting talk. I like that a lot. As a former oceanographer and an atmospheric scientist, I was intrigued on both fronts there. You had mentioned earlier that the claw hypothesis wasn't entirely dead, and that there is some correlation with, or some, I heard that you said there's some causal relation with DMS. And I didn't see you talk about that. And I could certainly see that there'd be a correlation with DMS because DMS is produced by biological organisms. And when there's high DMS, there might be other factors going on. But do you think there's a direct causal relation between the high CCN and the DMS? I'll leave it at that.
Okay, so all we have are correlations. So that's an excellent point.
Okay.
But the mode of aerosols so the transported aerosol, it comes from the transported aerosol is eliminated by radon, right? So we look then only at cases where there's no transported aerosol. And then you look at the size of modes of available aerosol. And the size of mode of variable aerosol is consistent with secondary organic aerosol. And then you presume, yes, and then you stop. And so instead of DMS, should have mentioned that you certainly could have other VOCs.
Okay.
But that's evidence that it's a VOC.
It's a VOC, yeah. Okay, great.
Yeah.
Thank you. Very, very interesting talk again. I liked it. Fun stuff.
Thanks.
Right. Anyone else? I have a comment or question. I mean, what you showed is for the warm cloud mechanism. In a way, for the modelers, it's good news. Or if they want to model clouds over the ocean and it's dominated by the salt, and they don't need to put so much attention to the organic, that would truly simplify the modeling of maybe the liquid phase and mixed phase clouds or so over the oceans. And I think I would have guessed that the salt is so dominated. There are some studies, not very much, I think Colin O'Dowd's group and others, who say if there are surfactants involved, strong surfactants, they could still change a little bit the activation, how should I say that, history of the sea salt particles. And so I'm wondering, you have tensiometer measurements as well, surface tension, it seems. So that would be quite an interesting insight when you would pick something with high surface tension. Maybe there you see a change in the activation curves or so.
Yeah, no, that's a really good point. And some of the work at Mesa is really interesting. Like I'm sure you're familiar with the high CCN, low hydroscopicity stuff. That's like dichotomy conversation. And that's right. And so that would suggest that is not what I'm saying here. And I think one thing I would say is it's tremendously useful to be in the middle of the ocean rather than on the coast. So that is one difference. And yeah, I mean, this is only one study, but the surprising thing or a disappointing thing in our surface tension measurements were you really saw almost all the surface tensions I think they were all statistically insignificant differences. Like there just wasn't any real change in the surface tension. So much so that we did the surface tension measurements in 3 places because we were working really closely with Rich Moore, and so we used NASA's tensiometer as well as our own, and as well as one in the engineering department here. So there's no trouble with that measurement. It's possible. For those of you who have sampled the sea surface microlayer, the sea surface microlayer is badly defined or loosely defined, and some say it's a few molecules or some nanometer of depth, and maybe it's an entire micron, or maybe it's more. And we were sampling more depth of that layer. So
well, it's
so in that way, I could have underrepresented the organics. But yeah, but what we're seeing is when they're there, they're just terrible at picking up any water and becoming droplets, and so that's why the salts. Yeah.
No, I mean, it was more a comment too. So it's a challenging measurement or doing that. Yeah. Well, any more questions?
Daniel, this is Malcolm. Can you hear me?
Yeah, we hear you, Malcolm.
Oh, Sarah, thank you for a very interesting and sophisticated talk. I'm a physical oceanographer, but I'm very interested in the topic. You know, you mentioned in the early days of the DMS work, this publication by Charleston, Lovelock, Andrea, and Wood. Is that the James Lovelock of the Gaia hypothesis?
Yes. And
what's the latest on that kind of wild concept he had? Is he still alive, and is he still working?
Wow, that's like 3 questions I'm not sure how to answer all at once. Okay, thank you for those. So based just on what we observed in names, the rest of the cloud hypothesis wouldn't be valid, in that the rest of that hypothesis was, once you've got cloud drops, it's the cloud albedo is actually regulating climate and regulating, so it's a cycle, and regulating the phytoplankton that are able to grow, and the idea is that organisms are going to grow more and make more clouds according to the temperature that they would want, right? And so while our evidence supports that process is happening, it's not the majority of clouds. So then if the phytoplankton were in fact, as part of the Gaia hypothesis, to be steering the situation, they'd have a really terrible steering wheel, because they have 50% in the winter and less the rest of the time. So it wouldn't be a way to actually modulate the climate effectively. That is only one piece of the Gaia of Lovelock's work, though, and I don't feel qualified to talk about all of the ways in which ecosystems might modulate climate. I can't do it. I'm sorry.
Anyway, it's interesting reading. Thank you.
Yeah, just to check here, I got a chat from Ernie Lewis. James Lovelock is still alive. He's 102 years old. 102 years old and still alive.
How old, sorry?
102.
102. Well, he's part of the living ecosystem of the planet.
Yeah.
So,
wow.
So thank you, Ernie, for that information. Well, then I would say, if there are no other questions, we conclude now. I invite the graduate students to stay on. Sarah as co-host and can take over. I will log off as well. I thank Sarah again for giving this nice talk.
Thanks, guys.
The name study, it's good to see that so summarized.